Hello everyone, this is Shangxian and today I'm going to talk about Thalamus segmentation using convolutional neural networks. To begin with, let's get some background of the thalamus. Thalamus is a very important part of the brain. It monitors the signals, regulates the consciousness and alertness. So here is a 3D model showing the position of the thalamus in the skull. Thalamus is associated with many neural system diseases such as traumatic brain injury and multiple sclerosis. Therefore, being able to calculate the thalamus volume is very important to quantify such disease. However, currently we lack a more accurate algorithm to segment thalamus on the MR image. There are some very popular algorithms, such as FreeSurfer. It takes the structural MRI as the input, such as T1 or different modality. It produces a parcellation of the thalamus. However, FreeSurfer is very slow and lack of accuracy. So here's the image showing you the prediction from FreeSurfer. Compared with the manual delineation, we can see it's pretty off. Furthermore, FreeSurfer only uses structural MRI, and it didn't use any other images such as diffusion tensor imaging. So, diffusion tensor magnetic resonance imaging, we also call it DTI, is an MR imaging technique to measure the diffusion of water in the tissues. It can also model the white matter fiber tracks, and therefore is very helpful for lesion segmentation and also for the study of traumatic brain injury. We can also get some feature images from the DTI. So this image shows you one slice of the DTI image. It is a tensor image. From the tensor image, we can get all the scalar images such as AD, FA, MD, and edge map. From these images, we can see the boundary of the thalamus is easier to see. And the random forest thalamus segmentation algorithm actually uses the DTI image and the MP ridge to segment the thalamus. It is a random forest based algorithm. So first of all, they are trying to find a bounding box. Then they would use the bounding box to crop the MP ridge and also the DTI feature images. Then we can get a segmentation of the thalamus. However, graphs is very limited on the dataset and it's very slow. So we want to update graphs and replace it by deep learning. So here is the framework of our algorithm. First of all, we will construct a bounding box. Then we will crop the images according to the bounding box. After cropping, we will feed images into the 3D network. After the prediction from the 3D network, we will do some post-processing to make sure everything is smooth. So to construct the bounding box, we use 233 MA Cruz thalamus labels. MA Cruz is a multi-atlas whole brain segmentation algorithm. Prior to MA Cruz, all the images are already aligned to MNI space. We extract the thalamus labels and sum and average all the labels. Therefore, we can get a probability map for the left thalamus and for the right thalamus. From the probability map, we can also get a bounding box around the thalamus. After we get the bounding box, we will crop all the images according to the bounding box. As we can see on the picture here, those images are cropped around the thalamus area. It makes the training much easier and faster. After cropping, we will put all the images into our network. The network will predict our thalamus label. So in this study, we have two data sets. One is used for training. This one has 22 subjects. Each of them has the manual delineation, the FA image, MD image, edge map, and MP ridge. For the testing, we have 10 subjects. They also comes with the delineation, FAMD, edge map, and MP-Ridge. Both images are processed using the standard neural image processing pipeline, such as registration and for correction. Again, the training and testing are from two different studies. The input to the network is a combination of MP-Ridge and feature images from DTI. We can see the picture here. The Thalamus mask is overlaid on the images. All the images are cropped to 96 by 96 by 96. The left and right thalamus are cropped separately, and the left is flipped to look like the right thalamus. We input all the cropped images into the network, and then we get a segmentation of the thalamus. For the training and evaluation, we used data augmentation to increase our training data. We used the dice coefficient for the loss. The network is trained for 350 epochs. The total training takes about 12 hours. And here are some of the results. From this figure, we can see that the left part and the right part are different slices from the thalamus. The top row is the ground truth, and the bottom row is the result from our 3D UNet. We can see that the UNet is able to capture most of the thalamus edges, although it's missing some small parts. We also did a visual comparison with RAFs and MA-Cruise. We can see that RAFs and MA-Cruise tend to include regions don't belong to the thalamus, but our proposed framework is better at capturing the boundary. We also did some numerical calculation where we found our dice coefficient is larger than MA-Cruise and RAFs. So in conclusion, we use a bounding box to crop images and then send them to the 3D network. We use DTI features that can help identify the thalamus. It is the first deep learning algorithm that uses DTI features and MP-Ridge to segment the thalamus, and it's able to outperform other algorithms in terms of accuracy and efficiency, and it also works well on a variety of the data set. So thank you.